What determines the fate of a cell? The notch signaling pathway. Every baby has the same set of DNA in their stem cells, yet these cells divide to renew themselves and to give rise to different types of specialized cells. Stem cells can divide to produce themselves and also to differentiate into cells like pancreatic cell, muscle cell, neurons, red blood cell, epithelial cell, and all other kinds of cells in the living organism. And the question is, what regulates these cells to develop into these specialized cells? The answer is the notch signaling pathway. Notch signaling pathway regulates cell fate determination by regulating proliferation and differentiation. So flies that are usually mutant for notch show notched or jagged looking edges on their wings. Notch signaling usually occurs between two adjacent cells. So one cell called the signal sending cell the signal sending cell, and this signal sending cell usually has a ligand called delta. And the other cell is called the signal receiving cell, which has a receptor called the notch receptor. The notch receptor consists of two major domains, the notch extracellular domain and the notch intracellular domain. Step 1 of the notch signaling pathway is interaction of delta from the signal sending cell with the notch extracellular domain of the signal receiving cell. Step 2. There is a ubiquitination of the delta by an E3 ligase. So the delta ligand is ubiquitinated. Also leads to the exposure of some amino acid sequences between the notch receptor. Step 3. This exposure also leads to an S1 cleavage by the enzyme family ADAM. This cleaves and releases the notch extracellular domain, which is bound to the ubiquitinated delta ligand. Step 4. Inside the signal sending cell, there is a pooling effect of the complex, the delta, together with the notch extracellular domain, and therefore this complex is endocytosed into the signal sending cell. Step 5. Inside the signal receiving cell, the notch intracellular domain is therefore cleaved by a second enzyme called the gamma secretase, which releases the notch intracellular domain into the cytoplasm, and this not intracellular domain is translocated into the nucleus, where it binds to transcription factors involved in activating target genes that regulate proliferation and differentiation. Delta ligand interacts with Notch extracellular domain, and the delta ligand is ubiquitinated, and together with S1 cleavage by the ADAM enzyme releases the delta Notch extracellular domain, and this is endocytosed. The complex is endocytosed into the signal sending cell, and in the signal receiving cell, a second cleavage by the gamma secretase releases the Notch extracellular domain, which therefore translocates into the nucleus to bind to transcription factors involved in the expression of Notch target genes, which ends up in regulating cell proliferation and cell differentiation. This will be all for today. Thank you. Before we go, we would want to talk about some few fun facts. A fun fact. So, the Notch signaling pathway was discovered in the year 1900s by a Nobel laureate called Thomas H. Morgan. Thank you very much for watching, and we hope you subscribe to this channel for more. Thank you. Bye.